Okay, this is the second lecture from Dr. Neil Cross on the Biomedical Investigations module. We're looking at genetic studies of cancer and specifically using molecular techniques to look at cancer subtyping and also look for minimal residual disease testing in cancer patients. So in cancer, what we want to do is use molecular genetic and cytogenetic techniques to tell the difference between normal cells and cancerous cells. And cancerous cells have, by definition, genetic abnormalities which differentiate them from normal cells. And these are sometimes visible, as in at the chromosomal level. We could get here's some normal tissue, and the chromosome here has become abnormal, a translocation or a deletion or some other abnormality. And if this happens as an initiating event, it is present in every single cell within the tumour. And if that's the case, we can use that as a diagnostic marker to detect and tell the difference between cancer cells and normal cells. Now, in other situations, these cytogenetic abnormalities could be very heterogeneous, so that one population of cells have an abnormality, but another population of cancer cells don't. And in that situation, these abnormalities are not so helpful. So it's the ones where it's a clonal abnormality that occurs in all of the tumour cells that's most likely linked to an initiating event in cancer. Those are the ones that are most useful for tracking cancer within patients. Now this slide shows a fairly typical karyotype from a cancer cell. In this case, it's a fairly rare, obscure tumour type called uveal melanoma, which is just a tumour type that I've worked on. And this is a karyotype from a tumour population that I've cultured in the lab. And what you can see is, compared to a normal karyotype, there are lots of different changes going on. So the way that we write out karyotypes is the number of chromosomes, what are the sex chromosomes that are present. So this was from a male patient. The numbers and letters are missing off the bottom here, but you'll be able to see them on the main PowerPoint slides. So this is 19, 20, 21, 22, X, and this is where the Y chromosome would be. So it's 54 chromosomes with gain of these chromosomes. There's some cytogenetic nomenclature here that denotes an isochromosome 10q. So that's two 10qs stuck together. That's a long arm of 10. And then some additional chromosomes added in there. So this is the sort of thing that you can see from karyotype. You get lots of gains of chromosomes, loss of chromosomes, and structural abnormalities. And this actually represents a relatively, I won't say normalish, but a karyotype which doesn't have an awful lot of changes compared to most cancer cells. It's a relatively simple cancerous karyotype. Now, in this particular tumour type, and we'll see this later, there are certain chromosome abnormalities that determine whether the patients are likely to live or die following their therapy. So in some cancer types, cytogenetic changes can predict how well the patients do in terms of survival. In other chromosome abnormalities, they can predict how well they respond to certain drugs. Okay, so last week you saw this particular slide, which I went through, which explains how fluorescence in situ hybridization is performed, which is a technique which has a better resolution than cytogenetics. So I'm going to be talking quite a lot about fish, so I'll just give you a quick reminder. So here's a microscope slide. Here's some chromosomes attached to that microscope slide. It could be in the form of chromosomes. It could be a cell nucleus from a formalin-fixed paraffin-embedded piece of tissue. Depends what sort of fish you're doing. But what we are doing here is using a green probe to recognize one particular chromosome and a red probe to recognize a different bit of a different chromosome. Then we counterstain all of the chromatin with DAPI, which is the DNA binding dye, and look down a fluorescence microscope, and we can count the number of signals, which allows us to give us a chromosome count. So in this particular example, it's a chronic myeloid leukemia. This is chromosome 9, this is chromosome 22, and there's 9, there's 22, and that is what we call a fusion. It's due to one bit of one chromosome sticking onto the other chromosome. It's a chromosome 922 fusion, and we'll see that in more detail fairly shortly. So this is shown here. This is a chronic myeloid leukemia. It was the first tumour type to be shown with a chromosome abnormality. So you can see the karyotype is a bit of a messy karyotype, not the world's greatest, but that chromosome 9 is a little bit bigger than it should be, and that chromosome 22 is a little bit smaller than it should be. And basically what has happened is there's been a translocation between these two chromosomes so that if we look down here, this is chromosome 9, this is chromosome 22, and what's happened, and I've just readjusted the size of the window to show you, this chromosome 9, the base of this chromosome 9 has broken off and attached to the bottom of chromosome 22, and the bottom of chromosome 22 is broken off and attached to the bottom of chromosome 9. And so this is what we call a reciprocal translocation. Now what it's done is it's brought this piece of DNA, which is identified by the orange probe, onto chromosome 22 next to the green probe. So you end up with a green and red or green and orange signal, which is shown here. So we can use this to confirm which cells within a patient's blood have got this particular abnormality. We can do that very quickly, and we can screen maybe 100 or 1,000 cells quickly down a fluorescence microscope and determine the frequency of this translocation by doing fluorescence in situ hybridization and quantifying the number of cells which have this particular abnormality to get a frequency in blood. So that previous slide was chronic myeloid leukemia, and this is from a solid tumour, again uveal melanoma. And these have been analysed by fluorescence in situ hybridization for chromosome 3 and chromosome 8. Now, the interesting thing about this particular tumour type is the chromosome 3 and 8 status determines whether the patients are likely to live or likely to die after they've had surgery. So in this particular situation, chromosome 3 is in red. You can see 2 reds and 2 greens in some normal cells, whereas in the tumour cells you can see that there's 1 red signal, so loss of chromosome 3 and then gain of chromosome 8. And we see this consistently. And it's the most common karyotype feature that we see in these tumours is loss of chromosome 3, gain of chromosome 8. And this gain of chromosome 8, you can sometimes get 3, 4, 5 copies of it. And what this shows here is what we call a Kaplan-Meier survival curve, and it's showing how long patients survive after initial surgery from 0 up to 15 years. And this is disease-specific survival as a proportion, so at surgery everybody is alive. And then in blue we have normal copy number, and you see that only a minority of those patients die from their disease. Whereas in green you've got 3 copies of chromosome 8, and then in red you've got 4 copies of chromosome 8, and you can see that the vast majority of these patients will die, and this is a highly significant observation. So this is a sort of situation where FISH can be used to very quickly give a patient a prognosis. So we call this molecular subtyping. And we can see this in more detail here. On the previous slide that was interphase FISH. On this slide here we've got a metaphase FISH, and if you remember from last week, a metaphase FISH is done where you get the tumour cells and you get them to grow in cell culture. So this is what's been done here. We've taken the cells from the patient, we've cultured them, they grow very slowly and the chromosomes are really badly condensed. So this would be really useless for a karyotype, but for FISH that's great. What we can then do is use something called a chromosome paint. And a chromosome paint is DNA from a single chromosome, or let's say lots of chromosome 3s. They've been isolated, put into one pot, and that DNA that's in that pot is labelled with a red fluorophore, such as Texas red, and then the same thing has been done with chromosome 8, that's been labelled green. We co-hybridize them onto metaphase spreads, and what you can see is we've got 1 chromosome 3 and we've got 5 copies of chromosome 8 in this case. So we would know that this patient is almost certain to not survive all that long following surgery. Now this chromosome 3 and 8 status is only relevant to this rare obscure tumour type uveal melanoma. Different tumour types have different chromosome abnormalities which may or may not confer prognosis, but I'm just showing you an example where it's clear cut. And then this is another example which is from one of my papers where we've been studying chromosome 3 and we've used a combination of karyotyping and chromosome paints to characterise a breakpoint on, in this case, chromosome 3. This is normal chromosome 3, and then this is chromosome 3 with a translocation. So there's something stuck on the bottom of chromosome 3 here. This is something other than chromosome 3 material, and that's proven by this fish experiment using a red chromosome paint, and we can see that when we get down to a certain point, the centromere is actually about there, although it looks like it should be there. And then all of this stuff on the bottom is from a different chromosome, probably a bit of chromosome 1, but we're not quite sure. This was just part of a study to try and map what's the important bit of chromosome 3 in uveal melanoma, but it's just illustrating the technology of metaphase fish here, metaphase fish here, and how it relates to a karyotype. And we can really use chromosome paints very nicely to show where breakpoints occur and sometimes find out where the rest of this bit of chromosome 3 has gone. In this particular case, it was just being deleted rather than stuck on a different chromosome. So we can expand that concept even further to what we call M-fish, or multi-colour fish, spectral karyotyping, 24-colour fish. It's all the same technique, and in that situation we're using 24 different fluorophores to identify all the 24 different chromosomes, and I outlined this briefly last week. It's really good for identifying chromosomal translocations in tumours where the chromosomes are really badly fragmented and it's very difficult to tell one chromosome from another. Now, to put it into context, this is a spectral karyotype of a tumour cell, a tumour cell. Doesn't really matter what it is. It's not uveal melanoma. I think it was colon, this particular one. But what you can see is most of the chromosomes have actually been either duplicated or damaged in some way. If you look at this karyotype, almost every chromosome is what we call acrocentric. It's got no shorter P-arm. It seems like the chromosomes have been pulled apart and chromosome arms have been lost, or the P and the Q arms have been separated. When we look at the M-fish, you can see that chromosome 1 is two big bits and two small bits. And here you can see chromosome 6. That is a complex translocation of 3 different chromosomes making up that remnant of chromosome 6. So there's some chromosome 6, there's some chromosome 7, and in that particular blue is chromosome 11. And you can see down here the couple of chromosome 9s, and then there's some chromosome 9 with chromosome 1 material on there. So there's no way you could figure that out from a karyotype like this. This is what the spectral karyotype looks like, and then when you line up the chromosomes, you get this. So it gives you some indication of the degree of chromosomal fragmentation. And this is more typical of cancer cells. This is how really badly damaged cancer cells often look when you karyotype them. Now, there's other ways which you can use fish-based techniques. One of those is to look at relative amounts of individual chromosomes. And one technique is comparative genome hybridization. And in this technique, we use it for tumours where we want to know whether a particular chromosome region has been lost or gained. And what we can do is take some normal reference DNA, in this case red. So we take some reference DNA, just some normal DNA, and we label it red. So we extract some DNA and use some DNA polymerase and some Texas red labelled nucleotides, or spectrum red, some other red dye. Label that DNA red, take some tumour DNA, in this case from a uveal melanoma, label that DNA green with either spectrum green or FITC, and then co-hybridize that DNA onto a normal metaphase spread. So this is just a completely normal metaphase spread, and we see where that DNA sticks. And if the tumour has got a gain of chromosomal material, then this hybridization here would tend to glow more green, whereas if the tumour has lost DNA, as in it's got a chromosome deletion, it tends to go more red because there's more reference DNA. And we'll put exactly the same amount, so we'll put on exactly 10 micrograms of this and 10 micrograms of this, mix them together, co-hybridize them onto a metaphase spread. And what you can see here is that the bottom half of this chromosome seems to be glowing green, and this one here is glowing green. This one's red, this one's green, and there'll be something else green somewhere, maybe it's not on there. And basically this is chromosome 11, this is chromosome 11, this is the X chromosome. So what we can infer from this is that this particular tumour has got a gain of chromosome 11q, because chromosome 11q is bright green, and it's got loss of either the X chromosome or the Y chromosome because there's less red DNA there. So there's more of a red signal, less of a green signal. And when we compare the karyotype of this particular tumour, it's the same tumour that we've just looked at on the previous slide. When we look at the full karyotype, you can see chromosome 1, 2, 3, 4 completely normal. Chromosome 5 looks abnormal. And the reason it's abnormal is because on the bottom of chromosome 5 there is a bit of chromosome 11q, which is this here. So this tumour has got trisomy of 11q, and it's also got trisomy of chromosome 10, and it's also lost its Y chromosome. This was a male patient, so we've got loss of Y. So we've only got one copy of the X chromosome, we've got 3 copies of 10 and 11q, there's the 11q bit, and when we look at the spectral profiles of chromosome 10 and 11q, chromosome 10 is above what we'd expect for a normal chromosome, like here. Chromosome 11q is raised, and the X chromosome is below what you'd expect. So the CGH, the comparative genome hybridization, is confirming what we can see on the karyotype. Now, comparative genome hybridization is quite an old technique, and that's been superseded by what we call array comparative genome hybridization, or just array CGH. And it works in exactly the same principles as comparative genome hybridization, but instead of hybridizing green tumour DNA and red normal DNA onto a normal metaphase spread and measuring the red and green ratio along each individual chromosome, instead you hybridize it onto a glass slide which has regions of DNA attached to it which represent individual chromosomes. So these arrays can have bits of DNA on them that represent hundreds of thousands of regions throughout the genome, and when you hybridize them together, if your tumour DNA is labelled green, reference DNA red, if some of these dots glow bright green, then there's an excess of tumour DNA at that particular location, and we know the location of that dot with reference to a particular chromosome. So in this case, in this particular example, it's been done with red as tumour. I mean, you can flip these round, it really doesn't matter. We often do an experiment red tumour, green normal, and then flip the experiment to prove that the result is correct. So in this result down here, we've got a tumour that's got a chromosome duplication down at the bottom of here of chromosome 8q24, that band. So, and that's a common site of chromosomal duplication. So the tumour DNA is labelled red, normal DNA is labelled green, mix them together, and then wherever these spots are red here, that actually represents chromosome 8q24, and then we get a spectral profile of all the different dots down chromosome 8, and you see there's an excess of tumour DNA at this particular chromosomal location. So it's a really good method for identifying chromosomal duplications, and in cancer we often get a small region of DNA, maybe only having a half dozen genes within there, but that bit of DNA gets duplicated again and again in cancer, which drives the cancer cells to grow much faster. So the key point to remember here, it's a CGH is a fish-based technique, this is an array-based technique, but the only reason you would use this is if you wanted to know the chromosomal copy number across the whole genome for a tumour sample compared to a normal sample, and that's what we do, and we get one of these red to green ratios across every single chromosome, and that will then home our attention to 8q24, which is the site of the MYC oncogene, for example. Now this is how fish can be used in more routine diagnostics. So everything I've shown you so far is on the verge of research informing diagnostics, but wouldn't necessarily be used as a primary diagnostic technique. The karyotyping for tumours is more research. The chromosome painting is done, it's a very odd example where the chromosome paints are done to assign prognosis. What is done very, very frequently is assessment of things like HER2 status in breast cancer. Now HER2 is a human epidermal growth factor receptor type 2. It's a gene that's overexpressed in breast cancer, and it drives highly aggressive breast cancer. And in about 20% of breast cancer cases, there is what we call a HER2 gene amplification. That basically means the HER2 gene is being copied again and again, and quite often the HER2 gene there are copies of it distributed throughout the genome. So you may have breast cancer cells which have got 50 copies of the HER2 gene. The HER2 gene, there's more copies of it, therefore you get more mRNA, therefore there's more protein, therefore you get more growth factor signaling. And in this example here, the HER2 gene has been flagged up in red by fluorescence in situ hybridization, and chromosome 17, which is where the HER2 gene resides, is in green. So we flag up the chromosome 17 centromere, and all of these red signals are the HER2 gene, showing that this is very, very highly amplified in breast cancer. Now, if we take a NAT tumour that we just looked at by fish and applied it to an array CGH, we would get a huge peak on chromosome 17 representing the HER2 gene amplification. So you could look for that particular amplification by array CGH, but this is a much more inefficient and expensive test than standard fish. But it could be done, and you would see a huge peak like this if you did that. Now, the fish experiment that I've just shown you tends to be done in concert with immunohistochemistry for the HER2 protein overexpression. And basically this is immunostaining for HER2 protein, and if these cells here, which have got lots of HER2 protein, are the sorts of cells that would have multiple copies of the HER2 gene. And if patients stain up like this, they are offered Herceptin, which blocks the HER2 receptor signaling, whereas if patients are negative and very weakly stained here, then they wouldn't be given Herceptin. And then the patients which have got moderate staining that's membranous would then typically be retested by fish to get a confirmation on that. And if fish shows that there's increased gene copy number, then they would get Herceptin. If fish was negative, then they wouldn't. And that is just summarised by this slide here. Now, fluorescence in situ hybridization is a great technique, but it's tricky. Working with fluorophores, fluorescent molecules, they are sometimes problematic. You also need a very specialist fluorescence microscope to be able to visualize them, and there are other problems with fish that I will come on to. So as a result of that, there's a new technique called chromogenic in situ hybridization, which works very similar to fish, but instead of having a fluorescent signal, we get a chromogenic signal. Now, the other reason this technique's really taken off is traditionally fish was done by clinical genetics laboratories, and I mentioned that in last week's lecture, that fish was a clinical genetic technique in the past, and then when cancer subtyping was required, because the clinical cytogenetics labs knew how to do it, they would receive the breast cancer samples and perform the fish. Now what's happening is that the histology labs that do the immunostaining that we've just seen, so the immunostaining like this is done by histology labs, histopathology labs, and they are used to doing immunohistochemistry. So if there was a fish equivalent that was like immunohistochemistry that would fit in with their automated analyzers, that would really go down very well with these labs, because fish done like this tends to be a manual technique, and it's a bit slow and laborious and usually done on a case-by-case basis. So this technique called SISH really helps because it's like a blend of immunohistochemistry with fluorescence in situ hybridization. It's much more amenable to be done in a histology lab. So this slide shows how SISH works, and you'll see how different it is to fish. So this in blue represents the target DNA, and this is the HER2 gene, and this is the chromosome 17 centromeric DNA here. And then in green represents the probe. In this case it's not fluorescent, it's labelled with DNP, a dinitrophenyl group, and in this case it's labelled with DIG, which is digoxygenin. Now both of these are what we call haptons. Those are small molecules that we can initiate antibodies to. So these are just small chemical groups, but we can generate antibodies that recognise DNP and digoxygenin. Now the reason that's really important is that we can then use an antibody to detect where the probe is bound. So previously we would have a probe directly linked to FITC and directly linked to Texas Red. Here we're going to allow the probe to hybridize, and then we're going to detect where the probe is bound with an antibody to DNP. So this is rabbit anti-DNP that's going to bind here, and this is mouse anti-deoxygenin, which is going to bind here, and then we're going with a secondary antibody linked to an enzyme. So we've got goat anti-rabbit horseradish peroxidase, which will bind to this primary antibody, which is attached onto here, and then we've got goat anti-mouse alkaline phosphatase, which is going to recognize this antibody, which is bound to deoxygenin. And then we can put on DAB to give a black signal, and we can put on fast red to get a red signal. Now this, once you've hybridized your probes, everything else is exactly the same as immunohistochemistry that the histology labs are routinely used to doing. Also, really importantly, that immuno can be done on an automated automated immuno machine that will do all the wash steps for you. And really importantly, the slide that you get out of the other end can be viewed on a standard pathology microscope that doesn't require fluorescence. So this means that SISH is really starting to take over from FISH as the main technology for looking for gene amplifications in cancer subtyping. We know that FISH is considered a better test, a better predictor of whether Herceptin works, but it's expensive, technically demanding, done on individual slides, usually done manually. Immunohistochemistry is easy, cheap, it can be automated, but there's subjective interpretation and issues related to fixation that's standard with histology. But SISH could take over from both of these tests, because you can, once you've done your initial probe hybridization, you can then treat the slide like a standard immuno, which the histology labs are very well used to. And most importantly, the signal that you get at the end is a slide that is viewed on a light microscope. If you view fluorophores on a fluorescent microscope, the signals fade as you irradiate them with UV, blue and green light to get the signals out, so the signals fade, whereas a chromogenic slide, the signals are fixed and permanent, and you could look at a SISH slide in 10 years time and it would look exactly the same. If you look at a FISH slide in 3 weeks time, it's probably faded to being not even visible. So the beauty of SISH is the signal is permanent, and also with SISH you get a much clearer idea of the tissue structure, whereas in FISH all you are viewing is the nucleus by DAPI staining. So with FISH sometimes you're not sure even if you're looking at tumour, because you can only see the nuclei, whereas with SISH you can see the shape and the outline of the cell, and you can be absolutely sure that you are looking at tumour material. Okay, in this last bit of the lecture I'm going to cover a topic called minimal residual disease testing. So everything we've looked at so far is taking a piece of primary tumour material, performing either karyotyping or FISH or CGH on that to get some sort of result. When we're dealing with cancer patients, one of the things we want to know is, have we got rid of all of the tumour, or are there still tumours cells floating around in the patient's body? And we can test this by doing something called minimal residual disease testing. And this is primarily done in hematological cancers, and hematological cancers are very amenable to this because they tend to have very simple karyotypes. So quite often, like we've just seen with chronic myeloid leukemia, and I'll just bring up the karyotype. This is chronic myeloid leukemia. The only thing that's visually wrong with this karyotype is the 922 translocation, which we can detect by FISH, which we can detect by karyotyping. So if that's the only thing that is visually wrong with those tumour cells at the chromosomal level, and it's present in every single tumour cell, we can use that to monitor how many tumour cells a patient has got at any one time, and does the tumour burden decrease during therapy. So that's what minimal residual disease testing is about. It's about monitoring tumour burden in patients during therapy. So this slide shows several different technologies that can be used to assess minimal residual disease testing in leukemic patients. So a typical leukemic patient at diagnosis might have 10 to the power 11 cells in their body which are tumorigenic. So at diagnosis, a significant minority or even a majority of tumour cells in a patient's blood will be leukemic, and that's when the patients are symptomatic. As the patients undergo either chemotherapy or whatever therapy they are receiving, once they get down to about 10 to the power 9, so that's just 1 billion tumour cells, they are clinically in remission and they are lacking in symptoms. They'll be feeling perfectly fine with 1 billion tumour cells floating around in their body. However, clearly, if you stop therapy when the patients feel better, the tumour will just grow back. So what we do is we carry on treating until tumour cells decrease and get to the point where they are as close to zero as possible. So we continue treatment until we are confident that the vast majority or all of those tumour cells have died. The problem is if you want to quantify those tumour cells, you've got really 4 different technologies: cytogenetics, FISH, flow cytometry and PCR. So cytogenetics, as we know, involves taking a blood sample, growing those blood cells in cell culture, and then doing a full karyotype on as many cells as we can be bothered to do. Now, I could karyotype 30 cells in the morning quite easily. However, if you're going to spend half a day on one sample and you only karyotype 30 cells, well, that means that by definition your limit of detection is about 3%. And if the patient's got 3% tumour cells in blood, half the time I'm going to see it in one cell, half the time I'm not going to see anything at all. So my limit of detection is 3%. So cytogenetics is quite rubbish. In contrast, FISH is much better. With FISH, I could take a blood sample, take out that blood smear, put it on the slide, hybridize probes to it, and then screen 1,000 cells quite easily in a morning. So look down the microscope and look at 1,000 individual cells, see what they look like, and if I see one of them that looks like this, we could say that that is 1 cell in 1,000 that's leukemic. So my limit of detection is 1 in 1,000, 0.1%. However, what we are doing here is looking down on a single cell, and every now and again chromosome 22 and chromosome 9 will be vertically in alignment, so that this green spot could be at the top of the nucleus and this red spot could be at the bottom of the nucleus, but they look like they are fused. So this gives you a false positive, and this happens every now and again. So every few thousand cells you look at, you'll get something that looks like it's got a fused signal when it hasn't. And if you only see it in 1 cell in 1,000, you don't really know if it's positive or negative. So we can also do flow cytometry, and this involves looking for CD markers on the white blood cell populations in blood. So, for example, B cells are typically CD138 positive. We could look for CD45, CD19, and basically there are loads of these different markers, and we know which ones should be present within blood. And if we see any populations of cells which have an abnormal combination of CD markers which don't normally exist in blood, they may represent a tumour population. We call this immunophenotyping. So flow cytometry is great in that we can perform analysis on typically 10,000 cells for a single run, but if we're immunophenotyping, we go much higher than that. And it means that if we do 100,000 cells and we detect 10 of them which are abnormal, we can get down to a frequency of 0.1% tumour being positively identified. And with the individual tumour types, we know which CD markers to look for. So that's a useful technique for getting the sensitivity even lower. But we're still down here of only being, even if we detect 0.01% of cells in blood, we're still at about 10 to the 7 cells in that particular patient. What we want to do is go even lower, so that basically if we take a 10 ml sample of blood and there's one single cell in there, and within that 10 ml sample of blood there might be tens of millions of white blood cells, if there's a single leukemia cell in there, we want to be able to detect it, and with PCR we can detect it. So the question is, how can we use PCR to detect a tumour cell? Well, we're only using it where we have a translocation, and the translocation has to be present in every cell in that particular patient's blood, and every tumorigenic cell in that patient's blood. So If we take a disease such as chronic myeloid leukemia, 90% of them will have this translocation between chromosome 9 and 20, which we call the Philadelphia chromosome, and it's a translocation between the BCR and the ABL oncogene. And these breakpoints vary between individual patients, but always result in what we call a fusion transcript between the BCR and the ABL gene. That means if we've got a fusion transcript, we can have one PCR primer in one gene, one PCR primer in the other gene or coding sequence, and perform PCR effectively across the translocation, but really only across one mRNA. And that allows us to detect the BCR-ABL fusion transcript by RT-PCR. So let's look at how we can use RT-PCR to detect translocations. So this is the BCR gene on chromosome 9, this is the ABL gene on chromosome 22, and they are stuck together by a translocation. And that translocation has occurred about halfway through the BCR gene and partway through the ABL gene. So this is the DNA sequence. BCR gene, ABL gene fused together. Now if we take this BCR gene, the promoter is here, and that will drive BCR mRNA, and there's introns, let's say intron 1 and 2 of the BCR gene, and then there's an intron, then a break in the chromosome, and then we're into an intron of the ABL gene, and then we're into a load of exons from the ABL gene. So basically the promoter of the BCR gene drives exon 1, 2, 3, the first few exons of the BCR gene, and then the last few exons of the ABL gene as from one mRNA. This is unique to the cancer cells. Now if we look at the protein that this translocation codes for, and this produces the P185, 185 kilodalton protein. This is the N-terminus, this is the C-terminus, and this just shows different protein domains. But basically, this protein here is BCR protein, and in this particular example that we get in chronic myeloid leukemia, this is acute lymphocytic leukemia, in this particular example, from this point left is coded BCR protein, from this point right codes for ABL protein. In all of these examples, this point onwards to here to the C-terminus is the ABL oncogene protein. So this is the last few exons of the ABL gene fused with the first few exons of the BCR gene, and that differs depending on where the breakpoint occurs. So what we can do is effectively amplify across the breakpoint by having one primer in one of the exons of the BCR gene and another primer in one of the exons of the ABL gene, and effectively amplify from here to here, but on the mRNA. So we're doing RT-PCR, and that gives us a PCR product which we can detect. And we can compare that to the total amount of ABL by having one PCR primer up here, one PCR primer usually in the same exon, or maybe the next exon along, so we can compare BCR-ABL transcript versus total ABL transcript, and that will allow us to quantify the amount of BCR-ABL transcript in a patient sample. We can perform quantitative PCR, and this, you should have covered the basics of quantitative PCR, but I can go through it in the tutorial, and I'll just realign the windows one moment. Okay, so we can now see the cycle number down here. So we're performing quantitative minimal residual disease testing, and what we are going to do is use quantitative PCR. And if you remember quantitative PCR, if you put in more target, you get an earlier CT value. Now a CT value is a cycle threshold. It's an arbitrary value. When we perform quantitative PCR, we measure the change in fluorescence detected per cycle. Go back and look through your quantitative PCR notes from earlier in this module, but we are using a forward primer, a reverse primer, and a probe that detects the PCR product that's generated. And we then monitor the reaction every cycle and then find the point where PCR amplification is detectable above background, and then at a certain cycle you will detect amplification that crosses an arbitrary threshold. This is where this red line is. So the CT value is the cycle threshold where the amplification curve hits that red line, which is about cycle 18. Now, Quantitative PCR is very quantitative. Here you can see the effect of diluting a sample, 20,000 copies, 2,000, 220 and so on, and you get a 3.3 cycle shift for every 10-fold dilution. We can go through the maths of this in the tutorial. So that's quantitative PCR. Is very, very quantifiable. What we can do is take a patient sample, measure the amount of BCR-able transcript before treatment, measure the amount after treatment, and we can find out how many fold change we have got in tumour burden. And if we say the patient starts off at cycle 19 and after treatment is at cycle 33, that's a 14 cycle shift. You should be able to calculate how many fold change that is, a fold reduction in tumour burden from pre-treatment to post-treatment. So we'll go through that calculation in the tutorial. If you've got it, if you can work it out, bring that answer with you and you will appear to be highly intelligent in the session. So what I'm going to show you here is how this quantitative PCR can be used to predict patient responses. So this is the predictive role for minimal residual disease testing in leukemia for BCR-able transcript detection, and that's BCR-able transcript versus total able levels in blood. Now you've got to remember that able protein will be there in low levels in normal cells. So what we're doing is looking at a ratio of normal cells to BCR-able positive cells and quantifying the amount of transcript of BCR-able to able. And if we find there's a high ratio, and this is only 0.02%, so it's still tiny amounts of BCR-able, but it's reliably detected at this level, and that predicts what we call a molecular relapse of 87%. A molecular relapse is basically a relapse from therapy, because in this case we are 5 months post stem cell transplantation. That's the treatment for the patient, and that should have eradicated every single BCR-able positive cell, and those patients should now have donor cells making up their white blood cells. If they still have BCR-able positive cells 5 months post-transplantation, they are almost certainly going to relapse, as in the leukemia is going to come back. So a ratio of greater than 0.02% gives an almost certain molecular relapse. It's a molecular relapse because it's a relapse before any clinical relapse. If you've got a low ratio of BCR-able, then you're looking at a roughly 50-50 chance of a relapse. Whereas if you're completely negative for BCR-able detection, so completely no BCR-able positive in a typical blood sample, then there's a 17% chance of a molecular relapse. And that could just be that for every 50 ml blood sample taken, there just didn't happen to be a leukemia cell in that 50 ml sample, but in the other 8 litres of blood in that patient's body, there could be a handful of cells still there, and they have the chance of causing a clinical relapse later on. So this is how the technology is used, and if you start to see persistent BCR-able detection many months after therapy, then you know that the stem cell transplant has not been entirely successful and we've got residual tumour cells, so these patients would go back onto some sort of anti-tumour therapy. So there's one other example where you can use a translocation to detect tumour cells in blood, and that's prostate cancer. So in prostate cancer there's this very common translocation, TMPRSS2 fused to ERG. The TMPRSS2 gene is an androgen-driven gene. It's a serine protease. ERG is an oncogene that drives the cancer growth. And this recurrent translocation is present in the majority of prostate cancer patients. So if we can detect this signal in patient blood, we know that there are circulating tumour cells in these patients. So I'll just realign the window so you can see all the detail. This is a positive control, so this is a positive control piece of tumour. This is patient number 1 and patient number 2. These are both patients who've had therapy. They've had the prostates taken out. They at this moment look apparently disease-free. But what I could do, and this is one of my analyses, could detect circulating tumour cells in both of these patients, showing that although the surgeon had removed the patient's prostate and they were on anti-androgen therapy, we still had tumour cells floating around in their blood. And this normal is a normal young healthy male. That was my sample. I was a normal young healthy male back then, and this is completely negative. So normal blood cells should not have TMPRSS2 ERG fusion, only the tumour cells should. So we can use this to monitor and track patients. We can take out people's tumours, but if we still see a persistence of tumour cells in blood many months after therapy, we know that there are still tumour cells floating around in that patient's body, and then we can put those patients onto more aggressive therapies. Okay, so this is a summary of everything we've covered. We've recovered fish. We learned about fish last week, but we've looked at it in the situation of doing a diagnostic and prognostic fish for uveal melanoma. Loss of chromosome 3, gain of chromosome 8 means the patient is very likely to die. We've also used fish in breast cancer, looking at the HER2 gene. If the patient has got high copy number of the HER2 gene, then that would result in the patient having a more aggressive tumour, but that is amenable to treatment by Herceptin. We could also do comparative genome hybridization. It's like fish. It's fish-based in a way. It's a type of hybridization technique. We can do CGH on normal metaphases. That's the old style way of doing this, to look for gene copy number changes along a chromosome. The more modern way of doing that is array CGH, where we hybridize normal and tumour DNA labelled red and green onto a DNA array and find out where that DNA preferentially binds, and that gives you your gene copy number across each individual chromosome. Then we've moved on to minimal residual disease testing. That's the detecting of tumour cells after therapy. And the most sensitive is qPCR, but we can only do that when we have a tumour marker that is very tumour specific. So if we've got a translocation that's unique to the tumour, and we can PCR across that or across the transcript from a fusion gene, that's very, very sensitive, and we can detect one cell in 100 million that way. Or you could do immunophenotyping, which is flow cytometry based, where you could detect one cell in 10,000, maybe one cell in 100,000. Fish is not very sensitive. You're looking at 0.1 to 1% of tumour cells, and karyotyping is very time consuming and very tedious, and no one would actually do that for minimal residual disease testing anymore. It was perfectly reasonable to do that 20 years ago, maybe 30 years ago. Then fish took over, then immunophenotyping has taken over for most situations, but where a translocation exists that you can PCR across, you would do this in preference. Okay, that's the end of this session. For the tutorial, I will go through any problems with quantitative PCR calculations and any discussions about these techniques and clinical situations where they are used. It might be a good idea to look back in your cell pathology lab as well, because there were references to HER2 staining in that particular lab, and these are very complementary techniques, particularly the HER2 fish.